So I would like to tell you about the work that's going on in the general feedstock genomics and genetics groups and how they tie together. So what I'm going to talk about is mostly genome biology and things that sort of emanate from the genome, so I'll give you an introduction to that, but it's clearly part of the cycle of development that we're working on at CABBI that starts from fundamental knowledge and basic mechanisms and then tries to integrate it with a design, test, learn cycle. The species that we're interested in are these rapidly growing grasses, and their phylogeny is shown here, just to give you a sense of the relationships. It's been about 10 million years or so since sorghum, sugarcane, and miscanthus diverged from each other. And that may seem like a long time, but on the grand scheme of things, they're pretty closely related. And so we can use a lot of information between the different species to try to inform one another. Just for reference, out there on the side is maize, which is also closely related. So we have a lot of information from closely related species. I realize I didn't have a slide for an introduction to DNA, which I think probably needs no introduction. Within each cell of a typical miscanthus plant, you have about 4 feet worth of DNA. So that's going to have some sequence. Everything that I'm going to show you here is dramatically simplified and representing the chromosomes, for the most part, as single units. But you have to remember that each of those units could be 100 million base pairs long. So what's shown here is a cartoon of the 19 chromosomes of miscanthus. Now, I'm going to talk about chromosome numbers. You don't have to keep all of it straight. But this number 19 is the number of chromosomes that is in a pollen, right? And so a typical somatic cell will then have twice as many chromosomes because they'll get one set from the father and one set from the mother. In this picture here, I'm showing you those 19 chromosomes, one from each pair of a somatic cell, and comparing them to sorghum in the middle. Do I have I guess I can use this. So the sorghum, there are 10 chromosomes of sorghum shown as these little lozenges in the middle. And then miscanthus actually has what amounts to 2 sets of chromosomes. These are not the mother-father set. These are a set that were produced by an ancient event where 2 different species hybridized with each other and left twice as many chromosomes. And we know that that's how it happened, by hybridization, because if I look at modern miscanthus chromosomes today, I find that half of them have a certain type of mark on those chromosomes that's left by transposon activity. And the other half has a different mark. And so when I look at these chromosomes, even though they're all chromosomes of the same species, half of them look like they came from one source and half look like they came from another. And that's the evidence that they came together by hybridization, which is a very interesting and fundamental process in plant and animal evolution. So that's one factor of 2, going from sorghum's 10 to miscanthus's 19, and there's a little fudge factor there because there was a fusion. There's another factor of 2, which comes from the maternal and paternal copies. So you all have 23 pairs of chromosomes, right? So the miscanthus corresponding number is 19 pairs. And so I really want to know the sequence of an individual miscanthus plant. I need to know the sequence of all 38 of those chromosomes. And now with new technologies, we can do that, and we have what I think are sort of spectacularly accurate and complete sequences now for several miscanthuses, and we're on our way to generating what's called a pangenome, trying to get a complete catalog or representative catalog of the variation that's present in the whole genus, in the whole, all the species that are available to us, thinking that that natural variation will be useful to us in the goals of CABBI. And so what's shown here is the mother and father's version of each chromosome in the 2 miscanthus references that we've already generated in collaboration with JGI. It's really hard to do justice to how, I think maybe you have to have been around for a long time to be impressed with how complete these are. When somebody says they've sequenced the genome, you think, okay, so now I know the sequence of letters from one end of the chromosome to the other. It's actually a much more tedious and annoying process. And over time, we've gotten better and better at it. And this is sort of, I have to kind of show you this. There were large regions that were missing from the sequence that we had generated that were very complicated and very repetitive. Now we've gone right through those regions. We really have a complete representation. What's next is, now that we have the ability to do this, we want to characterize the natural variation within Miscanthus. Miscanthus grows in East Asia. This is work that's, what I'm going to tell you is really from Eric Sachs and his longstanding efforts to understand this variation. And he's gone out, and he and his collaborators have collected samples from all around the different environments in this region. And so that's the complete variation within the species. If we could understand that, then we would be able to mix and match that variation to generate plants that had all kinds of different characteristics, even characteristics that in combination didn't exist in the wild. We're just getting started at doing this so-called pangenome, the multiple species. But one thing that we already know is that when I compare different individuals, or even just the maternal and paternal copies in a single individual, the chromosomes are not perfectly alignable to each other. There are large regions and small regions where sequences have been inserted in one chromosome or deleted in another that lead to variation that's more than just substituting one DNA letter for another. And that structural variation, it's a topic that we're interested in. Matt Hudson has done a lot of fundamental work in it. And this slide, which is probably more tedious to go through than is necessary, was a marker to me to mention this phenomenon. Of course, we're interested not just in the sequence of letters, but in how that plays out in the phenotypes of the plants themselves. And for that, we have to actually go to the plants. We have to stop working with just DNA and trying to link the DNA variation to the phenotype variation. And there's many efforts that are going on in Feedstocks group at CABBI to understand that, many of them emanating from work that Eric Sachs had done to prepare mapping populations. And I'm not going to go into that except just to give you one sort of early result of this. Now that these plants are out there, they're planted in the field, we know what the genetic variation is within the different individuals, we can start to look to see, can we understand for traits that we're interested in, like stem length and biomass, total variation that we're interested in. We can then look to see, do plants that have different genetic constituencies, genetic constitutions, produce recognizably different phenotypes. And this is work that's going on in collaboration with Ernie Siebert and Justin Yang and Brian Baldwin and their groups at Hudson Alpha and at AAMU and Mississippi State to try to understand how to do this. And we have very early results that are going to allow us to say, if you have this DNA variant, you are more likely to produce a particular phenotype. And it's that combination that's then going to be used in controlled breeding to try to accelerate that process. There's a kind of a basic science thing that has to happen before you do that, which is you need to understand in all of these different members of these large families that Eric has created, where the different pieces of DNA have been inherited from. And the basic phenomenon here is recombination. And this cartoon is meant to sort of remind you about this. Instead of showing you 19 pairs of chromosomes, I'm showing you just one, because that suffices to show the basic point. If I have a blue and a purple chromosome from mom and dad, when then I go to make my gametes, when the plant goes to make its gametes, it's going to produce recombinant chromosomes. And those chromosomes are shown here as I have a segment that's blue and then a segment that's purple. And the place where they cross over, the place where they exchange, is a place we'd like to understand in all these family members. So Therese Mitros, again working with Eric and other members of his group and Conchita's group, has produced, I think, a really spectacular genetic map that shows that the place where you're likely to get these changes, the place where crossing over happens and the color switches from blue to purple or purple to blue, are much more concentrated at the ends of the chromosomes. That's what this is showing. And that's a really fundamental point that's going to be a basic limitation in how we are able to do breeding for these kinds of traits. This is a slide I'm just going to show briefly. I like it because it grew out of one of these earlier CABBI meetings that we had in a discussion that Anna Hale and Eric Sachs and I were having, thinking about recombination, we were wondering about what was going on in higher polyploids. These are cases where instead of having 2 fundamental sets, 1 from mom and dad, you have 4 fundamental sets, 2 from mom and 2 from dad, something that can happen in plants and happens fairly commonly. And so the question is, could we engineer a system where we would have different chromosomes having different pairing partners? And I'm happy to talk more about this as a project that's ongoing now. All right, so once we have these genomes, we have the variation, we're starting to understand how that's related to phenotype in the macro sense. Here's the DNA, here's the plant. We'd like to be able to connect the dots. And this is work that's going on in the systems biology group. I have this nice slide from Amy Marshall-Colon that shows the interactions between understanding the genomes, understanding how the genes within the genomes are turned on and turned off, the protein products that they create and how they interact with each other, and then finally how the whole cycle produces the cellular phenomenon that are going to contribute to the organismal phenomenon that we're then going to break down and destroy and make fuels. To do this, we have to understand what the genes are, and a typical Miscanthus seems to have something like 45,000 genes, so way more than humans have, in part because of this doubling that I referred to. And Miscanthus, as much as we love it, is not a model system. We have to understand things in Miscanthus in the context of our knowledge of all other plants. And so this is a slide from Therese Mitros looking at the genes of Miscanthus and trying to compare them to the genes of all the other grasses whose genomes have been sequenced. We have about 20 of them. And it's this interaction that's then going to allow us to transfer information from well-studied maize, for example, into less well-studied, less well-understood Miscanthus, and accelerate that process of discovery and understanding. The ultimate goal then is to understand how these genes are interacting with each other. Here's another slide from Amy, showing that once you understand and have all these kind of integrated systems data sets, you can start to link the genes to each other and understand the flows of information as well as the flows of carbon and other elements in the cell, and then by virtue of that understanding begin to control them. And that control is in part going to be through breeding, as I mentioned earlier, from the large populations, looking at the natural crossing over, but now we don't have to wait for that, and thanks to Conchita and many others, we have the ability now to edit Miscanthus, so we can take variation that we've learned from our sequencing and insert it directly into a new context, and that level of control is kind of unprecedented. So we're always asked, what are we most excited about in the next 5 years? And it's always a balance between, am I really that excited about these small little things that we're doing, or should I make some grand pronouncement? And I think we're kind of in the middle, right? There are things that we're doing now that are going to be important in the long term. So we have now the genomes of a couple of Miscanthuses. They're wildly different from each other. If I look at Miscanthus sinensis and Miscanthus sacroflorus, 2 different species, their genome sizes differ by about 25%, much more than, say, human and chimpanzee, right? 25% in their length. How does that happen? What's going on? And how is that important, if it's important, in the phenotypic differences? We have all these genes. What's the networks of interactions that they play out? And in particular, how does that work at the cellular level, the different kinds of cell types? That's work that we're engaged in with the larger group. That'll then connect up to our phenotypic understanding, which we can do at an understanding level, or we can do just at a correlation level. And it's exciting to see these populations that are out in the field now yielding their information. And then finally, having this whole collection of genomes to see how that variation is going to be usable in our hands and in the hands of all of CABBI is exciting to think about. So thank you.